Hi, welcome to lecture three of our Science Saturdays lecture series here at Research Girl. If you're new here, welcome to our series. This summer, we're going to be hosting lectures every Saturday and sometimes on Sundays as well about a variety of different topics, including people's specific journeys in science, their research in different scientific fields, preparing for graduate school. So these are all free to attend, and we encourage you to please attend these lectures. They'll also be available recorded on our website in our lectures library. So if you want to attend not live, then you can do that as well. So without further ado, I'll let Ava take it from here.
Thank you so much. So my name is Ava, and this is my presentation, The Smallest Lab in Canada. Thank you so much for coming. So firstly, I just wanted to start things off with a bit of an introduction and tell you guys a little bit about myself. So as I said, my name is Ava Joa, and I'm 19 years old, and I'm from Vancouver, British Columbia in Canada. And currently I just finished second year in the health science program at Queen's University, so I am entering my third year. My research journey began in grade 10 when I started the Future Science Leaders program at Vancouver Science World. And it was a 3-year program that I completed, and it really opened my eyes to the world of research and really sparked a passion for research. And since then, I've pursued this research in both industry settings and academia, having interned at Acuitas Therapeutics as part of the vaccine development team and working as a member of the Cottrell Lab in the Department of Pathology and Molecular Medicine at Queen's. I'm also currently a research assistant at BC Children's Hospital with the Department of Rheumatology. Outside of research, I love to read, ski, cook, and when I'm at school, I play on an intramural inner tube water polo team, which is exactly as awesome as it sounds. So a little bit about my current research with the Cottrell Lab in BC Children's Hospital. So at the Cottrell Lab, my main project is working on profiling the tumor microenvironment in non-small cell lung carcinoma. So essentially what we're doing is we take tumor samples that have been provided us by Kingston General Hospital, and then we stain them with color-tagged antibodies, after which the antigens in the tumor light up and then allow us to quantify the area in the tumor that different types of cells make up. So it almost ends up looking like a paint by numbers. And using a super high-powered computer, we're able to take these extremely detailed photos of the tumor and then quantify the area that those different paint by numbers quadrants make up and get a way better understanding of the cellular makeup of different types of tumors. And yeah, so that's just what I was saying about conducting pixel analysis, which is the technical term for quantifying the paint by numbers. And it allows us to observe differences in immunotherapy efficacy for lung cancer patients at the cellular level. And so now at BC Children's Hospital, which is where I've been working this summer, I'm working on a project evaluating the clinical characteristics at diagnosis and 12-month follow-up for children with systemic and cutaneous polyarteritis nodosa, which is an extremely type of rare childhood necrotizing vasculitis, which affects small to medium arteries. And since it's extremely rare, while it is moderately well characterized in adults, there's really very little information about how the disease affects children specifically. So I'm working on a multi-center study that's been working with hospitals from around the world, and we have a cohort of about 50 patients at the moment. So, what was I talking about before? So, I'd like to get into Future Science Leaders a bit, which was a program that was so incredibly impactful for me and my journey in science. And I don't know if you've been following the World Cup at all, but you may have seen in Vancouver there was a giant building that was turned into a giant soccer ball, as you can see in the photo. That is Science World. And so it is a super amazing interactive scientific museum, and they have something for everyone, and they have rotating exhibits. But one thing that they have is they host the Future Science Leaders program, which is an after-school scientific development program for high school students, which is focused on developing scientific inquiry and laboratory skills. So the breakdown of the program I have on the slide right here. So in year one, students have the opportunity to learn from scientists in industry and academia on a variety of skills and topics. So it's really incredible that they have a lot of scientists who work in nearby industry or at the University of British Columbia who are able to come in and share their experiences with the students. And then in second year, you are given the opportunity to formulate an independent research project. You are given essentially total creative freedom on this. You are given a $100 budget and the opportunity to take 6 months to pursue a project of your choosing. And then in the third year of the program, 4 students from the second year are selected to continue their year 2 project was evaluating the impacts of synthetic necromones, which are necrotic pheromones, on insect feeding behavior and its potential suitability as a sustainable pesticide. So in the second year of FSL, I had no idea what I wanted to research, and all my peers were coming up with these really interesting projects, and there was so much excitement and so much passion around me that almost it made it more difficult to figure out what I wanted to pursue. And I ended up remembering something I had learned quite a while back on a trip to the Victoria Bug Museum, and that was when ants die in a colony, the living ants actually remove them and take them to a designated refuse area, almost like an ant cemetery. And the most curious thing was that after this ant takes the deceased ant to this graveyard, the other ants don't immediately let them return to the colony. And I was really struck as to trying to figure out why that was. So during my literature review stage, I looked into this constantly, and I was so curious. And then I found out it's because that there are specific necrotic pheromones released in order to scientists were able to distill the active ingredient in this pheromone. They treated living ants with it with a little paintbrush. And other living ants in the colony suddenly treated their sisters as no longer living, despite every obvious sign of life. And what really struck me most about that is if this pheromonal response was so powerful that it could override every sign of life, what else could it override? And so then, after coming to that conclusion, I really wanted to see how this could be applied and if it would be possible to have a pheromonal response that is so strong that it could override an insect's need to feed and forage. And so I was given the opportunity to work with cockroaches. They were very generously loaned to me by Vancouver Science World, and I worked with them for 6 months. But after my second year of the program, I was left with incredibly inconclusive data that left me really disheartened about my project. My data was all over the place, and there were really no conclusions that I could make for it, either for or against my original hypothesis. But then in the spring of my grade 12 year, I found that I was accepted for the third year of the program, and I somehow convinced my parents to let me take home 30 Madagascar hissing cockroaches that, as I said, lived with me in my basement for 6 months. So when I say the smallest lab in Canada, I am being extremely literal. I had a little bench that was just an IKEA table, a chair, a tank of cockroaches, one pipette, and a little scale for weighing out the chemicals I was using. And so now, after that, that led me to Acutus Therapeutics in Vancouver. And so that project really opened up a massive interest in science and scientific inquiry. And after finishing my first year of university, I decided that I really wanted to try and apply for an internship. And like I said before, one of the most valuable things about the Future Science Leaders program is that we were allowed to have the incredible opportunity to meet with so many scientific experts from across industry and academia and just have the opportunity to hear what they're so passionate about. And so, thinking about this, I began to reach out to scientists I had met through this program. And after quite a bit of emailing, I had the incredible opportunity to begin working as a preclinical studies intern in May of 2025. And as an intern, I was really bounced around through so many different projects. But my main kind of through line project was evaluating if an ELISA, which is an enzyme-linked immunoabsorbent assay, was a suitable replacement for the more expensive MSD analysis. So MSD analysis is almost it's extremely similar to an ELISA in what the actual kind of goal of the assay is. You're using both of them to quantify the amount of protein in a sample, which is amazing for when you're doing vaccine studies and you want to measure the amount of antibodies that in a blood sample to see if a vaccine is actually doing its job. However, the MSD plates, they're a lot more exact and technical. They're about at least $1,000 a plate. And so the scientists are really keen to see if an ELISA, while it is a little less precise, could most of the time be used as a general benchmark to see what vaccine candidates were already doing better than the others, and then could use that almost as a weeding process before more exact MSD research began. And so, as I mentioned before, I'm an undergraduate student at Queen's University in Kingston, Ontario. So, on the left, I have a little photo of my school. It's right on Lake Ontario. And then on the right, we have it on a bit of, sorry, a bit of a map, so you can just see where it is in approximation to other places. So not too far from Boston, and I'm not too far from New York, which is great. And so after my time in high school with Future Science Leaders, and after a really incredible internship opportunity with Acuitas Therapeutics, I knew that I really wanted to continue research in my undergrad, but I didn't really know what to do or how to get to this goal. So I decided to just email constantly. I know if you look online, you'll definitely find very mixed reviews about the efficacy and success rate around cold emailing, but at Queen's, that was really my only option. I hadn't taken any classes that had more of a wet lab component other than my first year chemistry course, and I felt very kind of in the dark about what undergrad research looked like, even though I had seen it. I had seen scientific research in so many other contexts. So I decided I made a list of professors whose work sounded interesting, and I just decided to email and email and email and email and just put myself out there however possible. And then I would have the satisfaction that, you know what, if things didn't end up working out, at least I knew that I tried my very best. And after about 20 emails and one interview from those 20 emails, I was given the really incredible opportunity to join a lab that works in between the Department of Molecular Medicine and Pathology and the Queen's Sinclair Cancer Research Center. So I began working with them in September, and I'm now given the opportunity to pursue an undergraduate thesis coming into my third year. So, as I was talking about the endless amount of emails I sent, choosing the right lab is extremely important, especially because while I was interviewing, I was questioned about why I wanted to join a more dry lab when all my previous experience was very wet lab facing. And so I thought I would just kind of break it down for you guys, because choosing the right lab can be so difficult because there are so many interesting things in science, but hopefully trying to separate your interests between wet and dry, at least in the moment, might help make this process a little bit easier. So when you're looking at the wet lab, the first thing I would say is that you learn a lot of skills that are really hard to learn anywhere else. You're extremely close to the experiment. When I was working at Acuitas Therapeutics, I was working in a biosafety cabinet with my arms in the bench and PPE on, and while it might sound a little daunting, it's really such an incredible experience that I would try and urge anyone, any young scientist to get involved with because it is just so amazing. And there was one time I was given the opportunity to help out with a human blood experiment, which I still don't really know how I got roped into that. And I was working with my supervisor, and she showed me the assay. She plated out the blood into a 96 well plate. And after doing it once, she turned to me and said, Ava, I don't want to do this anymore. You do the rest. And handed me these 7 vials of human blood. And what I love so much about wet lab work is there's always something different. There's always something new to try. And it's just super hands-on and really close to the science, which I love. However, the one thing about it is that the wet lab is very time intensive. Your experiments aren't exactly forgiving. if there's contamination, if something happens, you normally have to start completely from the beginning. And so that can be really daunting, especially when you're at university and you have your courses, you have clubs, there's so much going on already that I knew I didn't want something that was completely hinging on hour to hour success. And because of that, I was really keen on building my skills in a more dry lab environment, which focuses on computational data analysis. And as it says on the slide, data analysis is an incredibly valued skill and it's super transferable to so many other things. Um, while wet lab skills are super highly valued and generally pretty transferable, um, sometimes the experiments you're doing, and especially what kind of field of wet lab work, if you're working with animal models or tissue samples. There's a lot of different subskills, but dry lab, at least I like to think, is a little more cut and dry in what you're learning and then how you could apply it in a different scenario. I also find that dry lab work, there's definitely a lower barrier to entry because of how transferable these skills are. You probably already have quite a few dry lab skills that you might just not be aware of, whereas wet lab normally requires quite a bit more specialized training. The other thing I was really keen about when looking into dry labs is that the work is a lot more mobile. When you're working from a computer, you don't necessarily need to be in the lab first thing at the morning, especially because I go to school in Ontario and we had a couple -30 days this winter. So it's nice to not have to make it through the snow in time to have enough time to finish an experiment. However, the last thing, and I'm sure there's some people who are really going to disagree with this next thing that I say, because I know people are passionate about their computers. The dry labs sometimes can tend to be a little bit dry. There's definitely a sense of excitement that comes in the wet lab in being so close to the science. And having a couple degrees of separation in the dry lab can be a little less exciting for some people. However, I think that it really comes down to picking a topic that you're passionate about and something in science that you want to learn more about and that you're driven about, and that can really help close that distance. So, my number one biggest tip for any researcher, for any young scientist, is to get certified. And with the goal of reducing the amount of training that a lab has to provide you with. Because, in my opinion and my experience, this is something that makes you such a valuable candidate when PIs are looking at potential researchers to have join a lab, because you're just really cutting down on the amount of work that they need to do. And it's just a lot faster for you to be ready and actually get into the science. So a couple different certificates I just put as examples that I would really recommend. So one is, and just a note, as I'm Canadian, a lot of what I'm going to be talking about is the Canadian version of different certificates and different programs. So keep that in mind and search up the equivalent for whatever country you're in. But I believe there should be a ton of overlap, at least in theme, if not in name. So the first one would be WHMIS 2015, which is the Workplace Hazardous Materials Information System. And this is absolutely essential for any lab. And it teaches you the essential safety symbols that you'll encounter in a laboratory space. So you'll know how to tell your corrosive materials from your flammable ones. And this is an absolutely mandatory certification that you need to have in any laboratory space. And you can easily do this online. I did it through the University of British Columbia, even though I'm not a student. I was just able to register online and complete it. So this is something that you can pretty easily do on your own time, and I would absolutely recommend it because it is mandatory. The next thing I would really recommend is getting your chemical safety training out of the way. This is also essential and mandatory for almost every lab, and it teaches you to not only safely handle chemicals, but all the proper procedures should something go wrong, which is absolutely essential, especially if you're doing really independent work. And again, this is something I was able to complete online for the most part through the University of British Columbia. However, there was a 2 hour in-person practical where we were actually given the opportunity to show we knew our stuff and practice handling some mock chemicals safely. This is really going to change depending on what kind of research you're doing, but I would really recommend it because it opens the doors for what you're allowed to do, and that is completing your biosafety training. So the two that you're most likely to be exposed to are biosafety training level 1 and level 2. So level 1 is for working with non-pathogenic microorganisms like baker's yeast, whereas level 2 is agents associated with human disease like HIV or cancer cells. So I would absolutely recommend getting level 1 out of the way, as many wet labs might have that as a requirement if you're working with plant samples or anything like that. However, as I'm going to get this out of the way, I'm pretty biased, but as someone who loves working in a wet lab environment and being able to work with human samples like blood and tissue and human cell lines, I would absolutely recommend getting your level 2, as that again just opens up a lot of doors for what you're able to do. And similarly to chemical safety training, there was a very large portion of this that was completed online, self-paced, but there was a 3-hour in-person practical. So we could show in front of an instructor that we knew how to be safe in these types of settings. Lastly, I have the Canadian equivalent on one side and the American equivalent on the other, and that is being certified to work with any research involving humans. So, research involving humans is a very broad topic, especially in clinical research, because there are aspects of clinical research, especially if you're doing more qualitative studies where you are right in front of patients and you are giving questionnaires and talking to people. And there's also a side of clinical research where you're just working through pre-existing data sets, which is like what I'm doing. But either way, you need to complete this research ethics program, and it shouldn't take too long. It's not extremely difficult, but again, it just opens up so many doors for what kind of research you can do, and any opportunity where you are able to reduce the amount of work that someone needs to do, that just makes you a lot more attractive as a candidate and really incentivizes you taking you on the for the researcher. Sorry. Um, Kind of in a similar vein, one thing that I just really want to make sure that you know is you're a lot more skilled than you think. And while it might seem difficult to build scientific skills outside of a laboratory, please don't let that scare you off building your skills as a scientist or from recognizing the skills that you already have. Because as I was saying, especially in a dry lab context, you are definitely so much more skilled than you think, especially, so much about basic computer literacy is just such an essential part of our everyday lives. And so that barrier to entry is already just getting lower and lower as people's baseline knowledge gets higher. But three specific things that I just wanted to point out as examples are literature reviewing, scientific writing, and data analysis. So, even just to practice these things, or if you're looking at my slide and anything that you've maybe done in high school or university or any other kind of, or even on your own time, is just ringing a bell. I hope I can help you recognize that. But if you see something and you feel like you might not have a little bit of enough experience in that, there's so many opportunities to build these skills. Like with literature reviewing, if there's a topic that particularly interests you, practice creating a database of papers on Excel and break down all the different aspects, summarize the materials and the methods, and then the results and the discussion. And just really practice knowing how to break down a scientific paper so that in the future, if you need to do research on a topic, you know exactly where to go, and you don't waste time by trying to read the paper from the very top and getting stuck when it gets dense. Um, Other than that, try getting involved in a school journal club or work creating one with your friends or online. A lot of labs, I know my lab does, they have a journal club where basically the way it functions, it's almost like a book club, but for scientific papers, where once a week, someone is given the opportunity to do a mini presentation on a scientific paper they read that was really interesting. Everyone else reads it beforehand, and then you just are able to have really cool and engaging conversations on the topic. Next, I would really encourage developing your scientific writing skills. And a way to do this is find a student journal and submit to them with your own research, or see if there's any opportunities for you to become an editor for them. And becoming an editor, again, that really ties into literature reviewing and you have this process of determining what makes a scientific paper credible, what makes it well constructed, and then you also have that extra layer of experience working as an editor and being able to work with a scientist about how to polish a paper. And then additionally, many schools, one example that I picked in particular is Spectrum at the University of Alberta. They're looking for peer reviewers for these student publications. And so I would really encourage you to look at wherever you are in your own community and see if there are opportunities like this, because any exposure to practicing your scientific writing skills is just so absolutely essential, and it will make you more scientifically literate and more literate in general. And lastly, what I wanted to point out is data analysis. So much work in industry and academia relies on plain old Excel. So if you can really polish your Excel skills, if there are specific projects or data sets or anything you find in the world, there's a lot of amazing websites where you can find data sets. Um, in my first year of school of university, I took a statistics class, which is pretty common across many different scientific majors. But one of our projects was we just had to find any data set that we found interesting and put together a report on that. And anyone who knows me knows I am a massive movie buff. I watch a lot of movies and I love Letterboxd. So I decided to make a project parsing through my own Letterboxd data to see what was interesting, see what days of the week I tended to be watching the most movies on, see what months of the year I was watching the most movies, what genres, if there was any correlations. And basically the point of this little tangent is there you can find a data set anywhere. And if you can practice working with Excel, interpreting data, that is going to serve you so well in any career in science that you choose to pursue. And now I just want to talk about some really cool resources that exist. And one of them is LabPath. And while this is Canadian specific, I just wanted to mention it because maybe this will inspire you to potentially start something similar or seek this out wherever you are. But LabPath is, as it says, your hub for everything research in Canada. And it was meant to streamline the process of getting into university research as a Canadian undergraduate student. And so I just put together some photos from their website. So they have a really amazing university lab directory where you are able to filter by the school. So as you can see, I have Queen's, and then you can also filter by field of research. So I chose immunology, pathology, and inflammation, which is what I'm currently researching. And immediately it comes up with all these different professors who are currently recruiting for students, a brief blurb about what their research is, their email, and then you can see in the little green and blue blobs kind of what the intersection of their scientific research is. So I thought this was incredibly useful when I was sending all my emails trying to find space in a lab. And because otherwise that process can just really seem so daunting. And I found this to really help streamline the process and just make it a lot more manageable and approachable. The other resource I would really recommend, and again, this is a Canadian thing specifically. However, I am sure there are many equivalent resources and opportunities like this throughout the world. And this is the Undergraduate Science Canadian Case Competition. So this is a national research competition where students propose a novel research proposal based on existing research to solve a real world problem centering around a yearly theme. So I was able to participate in this past year's case competition, and the theme was reproductive health. So you are placed in a team of 2 to 4 other students, and you work on this topic and get to come up with a research question. And with the theme being reproductive health, we brainstormed and came up with the question of if main idea was why some individuals have very different reactions to hormonal contraceptives than other individuals do. And some people can have such severe adverse reactions, then that can really prevent them from utilizing certain contraceptive methods. And I was so curious about why there was that variability. And so our project was looking at, through the mechanism of insulin resistance, if socioeconomic status has any impact on a person's likelihood to have adverse reactions to hormonal contraceptives. So top scoring teams have the opportunity to present at the University of Ottawa to a panel of judges, and then all round one finalist teams. Round one being this written portion where you put together an abstract and submit it. Finalist teams then have the opportunity to have their abstract and potentially their entire proposal published in the University Natural Science Research Journal, which publishes research from students across Canada. So I just wanted to leave you with some final takeaways. And the biggest thing, I've said that a lot, the biggest thing, but I really want you to take this home, is to reject self-rejection. It is not your choice to make anyone else's decisions for them. There have been so many times where I felt like I was underqualified for something and that there were surely so many better applicants than me, but you have to put yourself forward anyways. Because once you put yourself forward, then whoever's hiring you, whatever position they're in, it's then their choice to decide if they think you're qualified for the position or not. You shouldn't try and make that decision for them. So show up eager, show up excited, show up audacious, show up without knowing everything. Just show up excited and just ask. You never know what opportunities might exist that you have no clue about. I speak from experience. I've had so many opportunities that just came about from me asking. There's so much that exists in science that isn't properly advertised, that isn't formally advertised. So reach out, cold email, and you never know what opportunities might exist that you just never knew about, or what opportunities could potentially be created. I've been super lucky to be able to stay involved with the Future Science Leaders Program as an alumni mentor, which was not a official position or really something that was existing before I started very eagerly emailing and asking if there was any way I could contribute. So I would just really urge you to be audacious with that and just really do your best to put yourself out there. And like I said, show up without knowing everything. Unfortunately, especially particularly within women, women are far more likely to not apply for something because they don't meet every single criteria. But as I said before, put yourself forward, show up regardless. And if you'd like a particular example, when I first began interning at Acuitas Therapeutics, I just finished my first year of university, as I said, and I had never taken a formal immunology course, despite um, wanting to work at a vaccine delivery system company. I didn't know very much, and there would be times where members of the preclinical team would be throwing out words and acronyms that I had just never encountered before. And I really had to learn how to work on to just kind of pick these things up and roll with the punches and just accept that learning is such a dynamic process. And I don't have to know everything at the beginning. I don't have to know everything at the end. But as long as I'm trying to learn more by the end than I did at the beginning, then it's successful. The other thing I would really recommend is stepping out of your assigned role because you never know who you might meet or what opportunities you might have access to. While I was working at Acuitas, um, I actually worked with several different people who were outside of my official supervisor, and those opportunities came from me messaging on the company Slack, just asking if anyone needed a hand, if anyone needed some data that they needed analyzed, or if anyone needed help in the wet lab, and that I was there and I was eager. And that led to, because of that enthusiasm, I was actually able to work so much further out of my initial scope that they had told me when they initially hired me. I got tons of opportunities to work with human tissue and try super different experiments than I ever thought I would have access to participate in. And as well, the most important people throughout your journey and throughout your life to surround yourself with are the ones who celebrate your crazy ideas. If we go all the way back to my time in Future Science Leaders, I remember telling the program supervisor that I wanted to work with cockroaches. And I still believe I'm the only person in that program who's worked with a live animal model. And I can remember his face very clearly when I told him that. He was just like, okay, you know what? If this is what you want, if this is what you're interested in, I will support you all the way. And even when, because I was working with live animals, he was forced to go to a cockroach husbandry training session, which involved him holding the cockroaches and learning how to care for them in his bare hands, which I know he was not keen about at all. He still really supported my project and supported my enthusiasm. And that's something that I've really noticed throughout my journey in science is that you have to find the people who hear your enthusiasm, who see your passion, and who see your weird and wild and wonderful ideas and just say, go after it, and I will support you. And again, probably still a cliche, but please trust in the process. There is so much that is in store in research that you might have no idea about at this point in time. I remember when I was in grade 10 and 11, and I was reading about the Future Science Leaders program through their website. And they did have a couple alumni give a statement about the program and kind of the things it's led to. And I remember seeing people talk about the $10,000 research grants that this program led to them achieving, or the incredible internships and um, postgraduate programs, and just all these really amazing opportunities that they all credited to this program. And I remember being on the bus, it was pouring rain, waiting at my bus stop, and I was just like, okay, yeah, sure, sure, this is, yeah. So at the time, I definitely couldn't see it, but now when I trace so many of the incredible opportunities that I've had through my journey, they really came back to that point of having a group of people who believed in me and who believed in my passion for science. And a lot of the most important work comes when you don't even realize that you're doing it. Um, every time I waited in the rain for my bus to take me to that program, I had no clue what it would eventually lead to. And so much of the stuff that is difficult and challenging now and seems like, why am I doing this? Stick with it. You never know what that might lead to. Um, but yeah, I just wanted to say thank you so much for having me. And if you have any questions, please feel free to ask.
Thank you so much, Ava. That was so great. It was wonderful to hear about your journey from, yes, the smallest lab in Canada to where you are now. And we're so thankful to have had you today.
Thank you.
If anyone has any questions, you can drop them in the chat, you can raise your hand, or you can just unmute yourself. We'll hang out for a couple more minutes just in case there are any questions.
I'm just thank you so much for your question. So I think in the case where research opportunities for high school students are limited, especially in fields like neuroscience, which it sounds like you're interested in. There's a lot that is available online if you're not able to find certain potential opportunities in person. I think, well, Research Girl is one of those opportunities, and just being able to form a community with other people who are interested in science, even if you have friends that you go to high school with who share a passion for science, if there's being able to continuously expose to yourself to a supportive environment like that is so essential and is really great for maintaining that passion and that drive. Um, I would really recommend working on a personal project. It's not only great for developing scientific skills and just maintaining that passion and your curiosity, but it's also just amazing to have that on your resume when you are potentially applying for different things in the future. So, like I was saying before, with putting together an Excel sheet, that was actually my first personal project that I was given when I was interning at a QS. They had me find every paper that I could on the applications of lipid nanoparticles in mRNA vaccines and create a giant Excel database on it. So if there's a particular neuroscience topic like that that really interests you, I would really recommend putting something like that together to just learn how to really read through a paper, and then also look through this database. And maybe there's certain conclusions that you might be able to draw? Is there a recurring limitation in different papers? And there's just so many great opportunities like that. Unfortunately, I'm not the most well versed in different international programs, but I would really recommend looking through certain things online that don't require you to be there in person. Like I was talking about the opportunities for developing your scientific writing skills and um, sorry, your literature, oh my gosh, literature review. A lot of those opportunities don't require you to be there in person. One thing I was looking at when I was looking at the Spectrum publication that they have at the University of Alberta in Canada, you're not required to be a University of Alberta student. You're not required to be a Canadian citizen. So things like that, through different universities across the world, can be really amazing opportunities. And then just a lot of what I found in science is there's kind of a web of recommendation. And once you work with someone, maybe they will point you towards a different opportunity, and then that person points you to a different direction. So I think just kind of trying to build that community for yourself is super essential. But yeah, but preparing for future opportunities, I would work on developing those skills and seeing if you can take ownership of a personal project and something that interests you.
Yeah, and I will add to that, since I have things to say on this topic as well. There are a ton of different organizations whose sole mission is to provide scientific research experience or education internationally and free of charge. So Research Girl, we are one of those. We have our traditional mentorship program that connects you with a mentor who will teach you about scientific research or help guide you through your own independent research project, or connect you with experiences and opportunities exactly where you are located or that are remote, so that you can practice those scientific research skills. We also have our independent research program, which lets you, regardless of where you are in the world, pursue your own independent research project. So something that I recommend you do, whether it's with Research Girl or on your own, is to do an independent research project, like Ava said. You can do a literature review, a meta-analysis, you can use archival data. There are so many ways that you can do your own research project without having to be affiliated with a lab or a university or have special access. There are a lot of open data catalogs that you can access, and you can actually publicly access data and use those as practice as well, if that's something that you're interested in, because that's the best way to start the gears turning with research, is to do your own research project and see how you take data and convert that into a result and analyze it and then discuss what those results mean. That's a big part of research that you can practice on your own without a lab. And then also at Research Girl, we have our journal, which anyone can submit to. So if you do pursue an independent research project, you can then get it published in our journal, so that you can get experience with the publication and the peer review process. And we also have, we're launching a free scientific research course this year called Protocol that is free for anyone to use. You get a certificate that says you have learned all of these skills in scientific research, and that will be available internationally to students. So that's also an option. And then similar organizations are things like the Youth STEM Initiative. You can also work with Academic Memories, is another partner organization of ours that lets you practice scientific writing and writing articles about science. So there are a ton of online opportunities that are available to anybody, regardless of where they may be located, and that are open to high school students. All right. So if that's all of our questions, I'm going to give like 30 more seconds. Yep. Okay. So if that's all of our questions, I think we will pause the recording. Thank you everyone for attending. And I hope to see you all at the next one next week. And I look forward to it.